Okay, now, so we've looked at the extracellular structures of the prokaryotes and how they define the overall structure of prokaryotic cells because they don't have much going on internally as far as overall um, larger subcellular structures. So let's work our way inward. All right, let's look at the plasma membrane. And what we learn about the plasma membrane, pretty much all the structural information is going to apply to eukaryotic plasma membranes too. And then we'll finish off with a few other components of prokaryotic cells. And then we'll move right into the eukaryotic cells and give you guys a brief overview of that. I know you've all had some intro biology, so I know you know the internal organelles of the eukaryotic cells. That's what we will focus on. But we're going to focus on the most important thing is to be able to compare the eukaryotic structures with the prokaryotic structures and know what is shared and especially what is different between the two classes of cells, right? So at the cellular level, make sure you keep that focus. All right, so the plasma membrane. So the plasma membrane surrounds the cytoplasm. That's why it's called the plasma membrane. So it's composed primarily of a phospholipid bilayer. You guys know that. We talked about that in the biochemistry section of chapter 2. Phospholipids. So the phospholipids form a monolayer spontaneously and then bilayers also spontaneously. Then they fold into enclosed structures, and they can basically separate an internal environment from an external environment, and that is the first and most important step in creating a constant internal, constant internal conditions that they want to maintain, which we call homeostasis, right? All right, so we have we have a phospholipid bilayer. All right, so here's a phospholipid bilayer down here. Let's see, here's the phospholipid bilayer. Okay, phospho. I'll put a phosphate symbol there. All right, P with a circle is a phosphate, in case you forgot. All right, and then we have the fatty acid tails. They're nonpolar and hydrophobic, and they create that hydrophobic sandwich, like a layer of butter in the middle of your sandwich. All right. And there are not polar heads, they're ionic, right? They are hydrophilic and ionic. They actually have full charges, all right? So this part is hydrophobic, okay? And the outer parts are hydrophilic. Now, within this part, we have some proteins that are attached at the surface. So we have some proteins that are all the way embedded. I'll talk about those in the middle. But either to the surface of the phospholipids, like around the heads, or to the surface of some of the embedded proteins, you can have what are called peripheral proteins. So they're kind of superficially attached, usually by ionic bonds. So there'll be some, negative charges and positive charges and matching charges on the proteins here. Okay, and they will match what's going on over there. Okay, so it's mostly ionic bonds. But then there's also proteins that are embedded in the membrane. And look at this. You guys recognize this structure? Little coils that line up next to each other. So these guys, they're called integral membrane proteins. Integral, they're integrated, they're embedded within the membrane. All right, and you could have integral membrane proteins that only go through partially through the membrane, like this. Right, if this was a protein. Let me try a different color. All right. Yeah, so here we have a partially embedded protein, but it's still an integral membrane protein. Okay. Now think about it. Whenever proteins are embedded in a membrane, the middle of that membrane here is hydrophobic between the heads, right, where the fatty tails are. So that means the amino acids on the on the part of the protein that crosses the membrane is going to have to be nonpolar, right? Hydrophobic to match. Okay, and as a pore, the pore is usually going to be hydrophilic because water and maybe ions have to go through, and they want to be create a friendly like versus like, right, environment. Here's another pore protein over here. This one on this side, it's got some alpha helices, as I was starting to point out earlier. Okay, lots of alpha helices, and they can create like a barrel, or so this could be like a channel or a carrier that moves things across the membrane, or it can just receive chemical signals and bind to them, and that binding will cause it to change shape and then stimulate all kinds of reactions inside. Get rid of that line there. All right. All right. So, and then, so these proteins that go all the way across, these are the integral membrane proteins. All right. Obviously, the ones that go all the way across are not just integral, but they're actually transmembrane proteins. Let me erase this. Boop. All right. Yeah. So these guys from the outside to the inside. And this is what's called a gated protein. It actually has a little piece of the protein that clogs it up until certain signals tell it to open. And then the alpha helices here, helping create a membrane protein. Here's just a long coil, probably some kind of helix and a peripheral protein and so on. We talked about that. So once again, the transmembrane regions need to be nonpolar. Remember my symbol, nonpolar, uncharged, not no polarity. Okay. So lots of nonpolar side chains, R groups, as they cross those membranes. But then on the aqueous side, so outside the cell where it faces water and inside the cell where it faces water, you're going to have lots of charges and partial charges, right? So basically hydrophobic as it crosses the membrane, hydrophilic on the protruding sides of the membrane. Let's look a little bit closer up here at the bacterial membrane. If you look really closely right in here, you can actually see the lipid bilayer. Can you see that? Can you see those two layers right there? So this is the plasma membrane. I'm going to label that again. So there's actually two layers here. If you look really closely, there's two layers. So this is a transmission electron micrograph, the ones that get highest resolution. Right? I can tell it's transmission because it has that thin, flat appearance, because it had to be microscopically sliced so you could see within the cell and get in close and look at stuff. So that's the plasma membrane. And then see this little thin, dark space line right here? The thin, dark line, that is the peptidoglycan layer of the cell wall, right? So it's very thin. Then this fuzzy structure out here, this is the outer membrane. And the fuzziness is the polysaccharides from the lipopolysaccharide layer that is present right there. And we know that if it has an outer membrane and it has a lipopolysaccharide layer, what do we know about it? What kind of bacterial cell is it? It's a gram-negative, right? Outer membrane, peptidoglycan, okay? And that also means that here's the outer membrane and the plasma membrane, and that space between, that's the periplasm. Right? So you actually see it. Pretty cool, huh? All right. So the ionic, I'm going to be picky about this. Yeah, so there's our ionic heads. They usually have full charges on them, okay, with phosphate group and then, glycerol backbone down here. That part's polar, but the rest of it's actually ionic. And then we have the tails. They can be saturated or unsaturated. We've talked about that before, those fatty tails. Okay, so that's any plasma membrane. So when we put together all the components of the plasma membrane, we have the hydrophobic. That's my symbol for hydrophobic. Hydro, and then Greek letter phi for phobic, not non-polar, same kind of symbol. All right, and we have embedded proteins, the integral membrane proteins. We have peripheral membrane proteins. You can have glycoproteins. There can be carbohydrates attached to the protein. You have glycolipids with carbohydrates. Carbohydrates, the sugars, are almost always on the extracellular surface, all right? This is the outside of the cell. So extracellular surface of the cell, intracellular surface of the cell, all right? The carbohydrates are almost always on the outside because they're for attachment. They're sticky. Remember, carbohydrates and sugars are sticky. And so they're on the surface so they can attach to other cells or other molecules um, or cells of the same type can attach to each other. But basically, remember the phospholipids, remember you can label them. I think I talked about this with you. For example, I could put a little, like a fluorescent molecule on one of these lipids, and I could watch it in the microscope over time, and you would see it diffuse randomly all over the surface. So these lipids, they are diffusing around side to side. They can't cross, right? I think I mentioned this. They can't cross, because that would mean you have to pull a very that ionic head through the hydrophobic layer. That is not energetically favorable. Okay. Um, Not only does the ionic head not want to go through, but remember the ionic head is actually playing with water, right? Water is excluding the tails to the middle, but there's a partial sphere of hydration around each ionic head, each phosphate head, and yet they would have to peel off that hydration layer before they could go through, and that's not energetically favorable. So that transverse diffusion, or what's called flip-flop, does not happen, not significantly. All right. So it's mostly lateral diffusion, right? Side to side diffusion in each layer is fine. And that's true for both the phospholipids and even the proteins that are embedded in the phospholipids, all right? Whether they are fully or partially embedded in the membrane, right? If we had a partially embedded integral membrane protein. But the ones that go all the way across, right, they're usually transporters or signaling proteins. They're called transmembrane proteins. They go all the way across, transmembrane. Trans means across, okay? But all these proteins and carbohydrates, glycolipids and glycoproteins, and there could even be cholesterols in there. We talked about that a little bit. There could be sterols between them. Remember in mycoplasmas, the smallest bacterial cells of all, they have lots of sterols in their membranes, and that toughens up their membrane, so they can survive and make it a little bit tougher, so they can survive without a cell wall. They actually just have a more lipidy, waxy membrane. So it's called the fluid mosaic model. So the lipids are very fluid, and everything embedded in them can kind of flow around pretty well. And the plasma membrane of your average cell is about as viscous and about as fluid as olive oil. All right. Is olive oil really thick and sticky? No, if you pour olive oil out of a bottle, it actually has to be pretty runny oil, right? But that's how viscous our membranes are. So they're very fluid. So if you had a little puddle of olive oil, you could poke a pin or a needle or something into it, and it might separate part of that puddle of olive oil. But then when you pull it back out, that puddle will seal back up again, right? Just gotta kind of push it in, bloop, all right, and then pull it back out, and bloop, it kind of flows back together, right? You can actually do that with plasma membranes or any kind of phospholipid membranes in cells. The proteins can diffuse around. That's usually part of their functions. They have to be able to diffuse around and move, all right? So it's essentially an ocean essentially of phospholipids with big proteins floating around in it, kind of like marshmallows in your hot chocolate or something like that, or ships in your sea, right? Or icebergs in your ocean, especially for the transmembrane ones, right? Icebergs go really deep into the ocean. So that's the fluid part because the phospholipids and their proteins flow around pretty freely. And it's called a mosaic because it's not just one thing, it's a bunch of things kind of mashed together, just like a mosaic art piece where you might use paint and tiles and I don't know, metal and wood and paper in your artwork, right? A mosaic. So that's the fluid mosaic model. That is the primary, the predominantly accepted theory or model. Model is just a visualization of a theory of how what the membrane looks like and how it works. And we can actually see a lot of it in the microscope, right? Electron microscopes. So the phospholipids, they rotate and they diffuse around laterally, and so do the proteins, right? So very fluid. So proteins afloat in a sea of phospholipids. That's how I like to word it. All right. So just like any plasma membrane, the prokaryotic plasma membrane has selective permeability. You might have heard the term in your earlier education, like middle school and high school, and I don't know, maybe even some college, semi-permeable membrane. No, that's not right. All right. So selective permeability is correct. Right. Semi-permeable means half permeable. Membranes are not semi-permeable. All right. That means they're permeable to about half of everything that comes along. That's not true at all. They are permeable to whatever they are structured to be permeable to, and that's it. All right, some things can get across randomly, either really small molecules or nonpolar molecules, because um, the double layer of fatty acid tails does not inhibit nonpolar molecules because they're hydrophobic themselves. They just slide right through. All right, but for most things, and most things are hydrophilic, they're polar or ionic. They need a protein to get across. Okay, a carrier protein or a channel protein, or even some kind of bulk movement. But bulk movement only happens in eukaryotes, so we'll save that for later. So in the membrane, some of the proteins embedded are actually enzymes, such as enzymes for ATP production. We'll learn about that in bacterial membranes, and also in eukaryotes in mitochondrial and chloroplast membranes. They have enzymes for making ATP and other sources of energy. But in what does happen, so remember that we don't get internal organelles in prokaryotic cells. But the plasma membrane, it can kind of bend in and make little bubbles, right? Um, and within these bubbles, This little folded, so this is your plasma membrane, right? You can have pigments embedded in there, and pigments are lipids. Um, yeah, a lot of pigments, at least photosynthetic pigments, are lipids, and they can embed themselves in there. So some bacteria have what are called chromatophores. So these little structures, they're called chromatophores, which just means colored structures, essentially, or color givers, I think is what it actually means. Um, so they're colored membranes. They have pigments. Okay, so here's some chromatophores, but notice they don't ever pinch off. If they pinched off into independent vesicles or something like that with contents, that would be internal membrane-bound organelles. You don't find that in prokaryotes, right? Um, so if they have sort of purplish or other types of pigments in there, then they are called chromatophores, and they're for absorbing light energy and using it for um, metabolic energy in the cell, for those that can do some kind of uh, photometabolism, all right? And if you fold your membrane in, and your membrane, so here's my membrane, and I'm going to fold my membrane in like this, so on and so forth. I'm going to put chlorophyll in there, not chloroplast, sorry, chlorophyll. So these are, once again, they're lipids. So these membrane pigments are lipids, so they embed themselves right into the phospholipid bilayer. And that chlorophyll absorbs light energy for photosynthesis. These are called thylakoids, which literally means uh, thylakos means little sac, and thylakoids are little sac-like things, right? Anytime you have oid, it means sac. It means it's like it. Like a humanoid is like a human, but not quite. So a thylakoid is like a little sac, but it's not a perfect little sac. So they're little sacs, and they have chlorophyll, and they're for photosynthesis. All right. So that's some special things that you can do with your. And so they're not allowed to have fully independent organelles, but they're allowed to have little folds that help store pigments for some level of photophosphorylation, right? Which is using light energy to make ATP, which we'll learn about later. What else was I going to say here? Hmm. Oh, so why would you fold the membrane? Why not just put yeah, the pigments are going to be in the surface part of the membrane too out here. So why would you fold the membrane? Why do we fold membranes? That's something that happens a lot in biology. Yeah, because when you fold it, when you have more textured surface, right, you increase your surface area. All right. So if there's more space, you have more room for more chlorophyll or other pigments, and so more room for absorbing light. Cool, right? Surface area is a big deal in cell structures and cell interacting with its environment. All right. So these membranes are barriers, but they are selectively permeable, right? They allow some things to get across, but other things don't. Mostly things don't, at least not easily, not directly. Okay. So what governs that permeability? Well, there are two major types of transport. There's what's called passive transport, right? You know what passive transport is? So in passive transport, like say we have these little molecules trying to get across our plasma membrane here, okay? So we have a bunch of them outside the cell, and we have some, but fewer of them inside the cell. Now, which way are they going to tend to move if they can cross the membrane? Yeah, well, they already show you, but right? It's going to go from an area of high concentration, all right, of that, assuming this is a solute, all right, to low concentration, all right? So high solute concentration and move to that area of low solute concentration, okay? So whenever there's movement, there's energy involved. Do you understand that? Whenever there's movement, there's energy involved. And so in passive transport, where does the energy come from? Because I've heard a lot of people say like passive transport doesn't require energy, and the other one, active transport, requires energy. That's not exactly true, right? In fact, it's not true at all for passive transport. It does need energy, but where's the energy? Well, when there's a difference in concentrations in one place as compared to another place, what do we call that? A concentration gradient. All right. And that is where the energy is. The gradient is energy. There's a difference in concentration. There's more on one side than the other side. The high concentration is a high energy state because there's more molecules and they're colliding. Molecules are always moving unless they're in absolute zero, but they're colliding with each other and repelling each other, and they want to move away from each other, right? Things always want to scatter. Things want to separate. Things want to move towards a state of chaos, all right? What do we call chaos or disorder, right, in science? So one of the laws of thermodynamics is things tend towards a state of entropy, right? And part of being entropy is not being organized, right? So when you have a lot of these molecules together on one side, that's too organized for nature to like. And they're going to repel each other and just want to get out of each other's way. So they want to spread out and spread out with other solutes in the solvent as much as possible. That's a less ordered situation, right? So the gradient has the energy. Just keep that in mind. So that's simple diffusion. So simple diffusion, they go across all by themselves. What kind of things can move across a membrane by themselves? Well, things that are either very, very small or things that are very nonpolar, right? And therefore hydrophobic. Okay, they can just kind of move across by themselves. So an example here might be oxygen gas, right? Very small. And oxygen gas is nonpolar, right? Because it's two oxygens bound to each other, so they have an equal fight going on. There's no polarity, right? Even though it's oxygen, it's fighting with another oxygen, so it's still an equal nonpolar bond, double bond between them. So that's one example. Facilitated diffusion. So facilitated diffusion, that's when things they're still going down a gradient because this is still passive transport. So we're still going down the gradient, all right, down the gradient. But these are things that cannot cross the membrane by themselves. So they're either too big and or too charged, either partially charged or completely ionic, right? So polar or ionic and too big. Now water is polar, but water is small enough that it can actually slide across, right? So water actually sneaks through. I've mentioned that before, okay? So water can kind of sneak through by simple diffusion. But if you want water to move faster, which we do in our kidneys and some microbial cells do this too, they put in water pores in their membrane that'll help water go through if you need water to go through faster, and these are called aquaporins, right? Aquaporins. Porins, if you can't read that very well. So there are carriers, like the one shown here. Now the carrier binds to a specific molecule on one side of the membrane. And look, it's at high concentration, right? Remember, brackets are concentration, right? That notation. High concentration on one side, and there's low concentration on the other side. So in this case, it looks like a sugar, right? It's a little hexamer. It's a hexose. Trust me, it's what they're supposed to be showing you. And now it binds here, and what happens is, this is important, everyone. This is really, really important. Whenever something binds to a protein, proteins have very complex structures, very specifically folded structures, all right? And if you have a very specific structure, it means you have a very specific function, okay? This is true for all proteins, because pretty much all proteins have a lot of specificity because they have a specific role in a cell. So something binds there, and in this case, it's going to be like a sugar, and we're going to have hydrogen bonding groups, hydrogen bond acceptors, hydrogen bond donors. They're going to interact with that sugar. But here's what happens, everybody. This is the coolest part, I think. Once this molecule, this transported molecule binds, the amino acid, the polypeptide chains move, and then the molecule to be transported is going to move down a little farther. Then the chains rearrange a little more, and it moves down a little farther, okay, until it gets all the way out, just like it did here. And then everything, all the chains move back into place, and it's closed, okay? So a carrier protein is always closed to the outside. And they bind really tightly to what they're binding to, okay? So any substance, any small molecule that binds, listen, any small molecule that binds to a protein to be transported is called a ligand. All right. Ligand just means a small molecule that sticks to a larger molecule, usually a protein. So it comes from the Greek word ligare, which means to stick. So a ligand is anything that sticks to a protein, and it's usually very specific, right? Like it says right here. There's a lot of specificity with proteins. They have very specific amino acid sequences of very specific amino acid that bind glucose or whatever sugar it is and move it through here. This looks like a glucose. Actually, it is a glucose transporter. Look, it's glucose. I didn't notice that before. All right. Um, but there's also, like I said, there's also channels. They are also passive transporters, and you go from a high concentration to low concentration. Even water can have a high concentration. Okay. If you have less solute, you're going to have more solvent, more water, right? And water will move through. But so a channel just makes a space of the right size and the right charge, right? Like over here, if I wanted to make a channel for like sodium, right, a sodium channel, I'm going to have hydrophobic proteins, right, in the transmembrane region. Same thing with this glucose transporter here. Non-polar proteins, but lots of polar proteins on the surface, and so on and so forth, right? Same thing here. And if you want sodium to go through this central space right here in the middle, what are you going to have to do? You're going to want to attract it, so you're going to put in how about some negatively charged amino acids, right? That's going to pull sodium through. So the right size and the right charge is going to pull it through, because you have to have the right structure if you need to do a certain function, all right? So we have channel proteins, which is like an open pore, the right size, right charge, right shape, for something to just glide through, go through that open tunnel. And we have carrier-mediated transport, where they have to rearrange the chains. So which one's faster, do you think? Channels or carriers? Channels, right? Because it's just an open tunnel and things just glide through pretty quick, right? So if someone opened a big tunnel and you guys are waiting to get into Levi's Stadium to go see the 49ers play. Okay, I live really close to the stadium. To be honest, my kids used to play soccer right next to the stadium. It's there's a soccer park right there. Um, and basically, if you opened a big tunnel, open these big gates, and there's just a big open tunnel, everybody could run in. It'd be kind of chaotic, but you get a lot of people in pretty quickly. All right. But what they usually do is they open the gates, then you have to, they have a few of them, but one person at a time has to go through the turnstile, right? Step through, turn, step through, step in, turn the stile, step through, step in, right? So it takes you a little bit of rearrangement and movement. So the turnstile is more like a carrier protein, and the channel is more like a big open tunnel, all right? So the channel is faster, but the turnstile gates basically they control things a little bit better. Now, which one do you think is more specific about what goes through? Like the sodium channel, right? It's had to be the right size and the right charge for sodium to go through. But maybe some other ions could go through too, about that they're the similar size or even smaller than sodium. But the carrier, it had to bind it, right? It's much more intimate. It has to bind it and rearrange specifically in response to that specific ligand. Okay, does that make sense? So these guys are fast, but these guys are more specific. Got it? Because there's a more intimate interaction. All right. Now, besides solutes moving across, like ions and sugars and things like that, like I said, water can follow too. You can either have aquaporins that allow water to move through quickly. Here's an aquaporin, right? Or you can have water can just move through by simple diffusion, because water's, even though it's polar, it's small enough to slide through the membrane pretty quickly. Not real easily, but it gets through. Okay. So, yeah, selectively permeable membrane, moving water. So when we move water inside, water also moves from an area of high concentration to low concentration. But now we're talking about the solute moving, not the solvent. Oh, excuse me, the solvent moving, not the solute. I said it opposite. So here we have some big solutes, right, inside of this permeable bag here. It's probably a dialysis bag. I work with these in the lab sometimes. So all these big solutes are in there. And outside, see, there's no solutes outside, doesn't look like, all right? So I'm going to say this is 100% water on the outside of the bag. But inside, there's a lot of solute. So let's say on the inside, of the bag, there is 5% solute, okay? And if there's 5% solute, what's the water percentage? What's the solvent percentage? 95%. All right. So where is water higher? Inside the bag or outside the bag? Outside the bag, right? Outside, inside. So which way is water going to go? Water is going to go in. All right, there's two ways to think about it. All right, and they're both correct. So think about both ways. Always use your brain in as many directions as you can, right? So when water moves in, it's going to keep moving in until the water pressure that's towards the walls of the bag is equal with the amount of pressure that's trying to push in, right? So there's two things that happen. One is that water is it's attracted, drawn in by solutes. But the other thing is that water, it moves down its own water gradient. Its own grade, okay? Moves down its own gradient. So both things are happening. Water is drawn in by the solutes. It's attracted to the solutes, but it's moving down its gradient, okay? And when it moves in, because water moves in, it puts pressure on the walls of the bag. And you think of the bag like a cell. If the cell doesn't have much support, like it doesn't have a cell wall like animal cells don't, it's going to pop the bag, right? So animal cells just they're really sensitive to osmotic lysis. So this is osmotic pressure that's going to build up inside the bag, OSM. Increase in osmotic pressure, right? And we know there's osmotic pressure because see, when water starts moving into the bag, you see the water started at the same level as outside the bag, but then water moves in and it moves down, and it actually creates enough osmotic pressure to push the water up in the tube against gravity and against atmospheric pressure. Okay, so that shows that the osmotic pressure is real, and it could definitely generate a lot of force inside of a cell and lyse that cell if the cell doesn't have a good wall. So you see why microbes and plants and fungi, well, fungi are microbes too, I guess. Fungi, algae, and plants have cell walls, and bacteria, right? So high water concentration to low water concentration, that's osmosis. And that osmotic pressure is real, and cells need to be able to deal with that. Animal cells deal with it because we have organs that regulate the osmotic pressure around our cells to keep an osmotic balance inside and outside of our cells. But if you're a single-celled organism or a plant, you just kind of have to deal with what amounts of water or salt or other solutes come around. And so they have cell walls to resist too much osmotic pressure, but the cell walls also keep the cells from collapsing if osmotic pressure draws water out, right? The opposite. So let's see how that affects cells. So the way that osmotic pressure affects cells is called the tonicity, all right? And so when we're talking about tonicity, we have isotonic solutions, we have hypotonic solutions, and we have hypertonic solutions. When we're talking about tonicity, we are talking about the properties of the solution outside of the cell. So in the last example, we're talking about the properties of the solution in the beaker, not the solution inside the bag, okay? So if you have equal concentration, if your solute concentration outside, so that's my solute S, if your solute concentration outside of the cell is equal to the solute concentration inside the cell, then it's the same in and out, and so we use the word isotonic. So same tension or pressure on the membrane inside and out, and so the cell's fine. All right, that's well balanced. The cell doesn't have to do a lot of work because everything is already homeostatic. Yay! All right. Now there's hypotonic solution. So hypo, right? Like if you're hypoglycemic, that means you have your blood sugar is too low. All right. If you have hypotension, it means your blood pressure is too low. All right. So your blood's not circulating well. But hypotonic means that there is low solute concentration. So the solute concentration outside the cell is lower than the solute concentration inside the cell. And so just for example, let's say that our solute concentration outside the cell here is 2% solutes, whatever that is, salts or something. And inside the cell, the concentration is 5% solutes. Okay, where's water going to go? It's going to move from an area of higher water concentration, 98%, to an area of lower water concentration inside, 95%, and it's also going to be attracted to the higher level of solutes. So water is going to move in, like it's showing here, and if your cell wall is not strong enough, boom, right? You get osmotic lysis, cytolysis. Cyto, which means cell, lysis. So breaking or splitting of your cell. All right, you don't want that. Okay, animal cells are very, very susceptible to that. That's why if you're putting fluids into somebody's bloodstream, you have to make sure it's isotonic, or else if they're hypotonic, you'll burst their blood cells. If they're hypertonic, the solutes are too high, water is going to move out. It's going to shrivel up your cells. And cells that are shriveled up don't work very well because all the proteins and nucleic acids and carbohydrates, they fall out of solution. The membranes shrivel up and they don't transport things right. And they just lose all kinds of osmotic balance, homeostatic balance, and they die. All right. So these guys, they will tend, if they don't have a cell wall, well, either way, they will swell. And if they don't have a sturdy cell wall, like if you expose them to, if it's bacteria and you expose them to like penicillin or something that weakens their cell wall, then they might even burst. Okay, it depends on the cell type. If they have a wall, they usually won't burst, and it's not easily. And then finally, we have the hypertonic solution. All right, if you have hyperglycemia, you have too high a blood sugar. All right, it's one of the symptoms, excuse me, of diabetes, for example. Or if you're hyperactive, then you're a little bit too active, you're a little spazzy. You need to sit down and, focus a little bit more. So hypertonic solution has a very, very high solute concentration outside. Okay. So the solute concentration outside of your cell is higher, or even much higher, greater than the solute concentration inside the cell. And if that's true, water, the net movement of water is going to be out. And if that happens, as you can see, as they show you here, it's not the best drawing, but as they show you here, the plasma membrane actually is usually attached to the cell wall. So you should see little attachments. But otherwise, the cell is going to shrink away from the cell wall if you have a cell wall. It's going to shrink away and shrivel up. And like I said, it's going to mess up membrane transport. So your homeostasis is going to go out of whack. Your proteins are going to dry up, and your proteins and nucleic acids and carbohydrates are going to dry up, and they're going to fall out of solution. They're not going to work right, and your cell's going to die if it stays like that very long. But when it shrinks up, if it just shrivels up into a little ball, like if it doesn't have a wall, then we call it crenation. That's shriveling up with no wall. But if you have a wall, and basically the we need a darker color. Here, let's try this. Try dark red. So if you have a wall, but your cell is shriveling up, even though it's pulling away from the wall, but it's still partially attached to the wall, this is called plasmolysis. So plasmolysis, it sounds like it means breaking of your cytoplasm, but that's kind of what it means literally. But what's really happening is your cytoplasm is shrinking up away, right? So the cell is just shrinking away from the wall. All right, so everything's becoming dehydrated, and your cell molecules stop working well, and your cell's not going to survive too well. Okay, so isotonic, everything's hunky-dory. Hypotonic, it's a lot of outward stress on your walls. Hypertonic, that's going to shrink and shrivel up your cell. All right, so osmosis, a simple diffusion, facilitated diffusion with a channel protein or a carrier protein. Those are all from high concentration to low concentration, whether it's a solute in passive and active, simple and facilitated diffusion, or if it's simple diffusion or even facilitated diffusion through an aquaporin of water, which is osmosis, it's high concentration to low concentration of whatever is moving. All right. But now there's active transport, because sometimes you need a lot more of something in your cell than outside your cell, or vice versa. And active transport, remember that even passive transport requires energy. Where does the energy come from? It came from the gradient. But what if the gradient is working against you? Okay. So since the gradient released energy to allow things to move down the gradient, if you're going against the gradient, it's not going to release energy. You're going to have to invest energy to make it happen, right? Rolling a ball downhill, that's passive transport. It just moves by itself. Rolling the ball up the hill, that's active transport. You have to put a lot more effort into it. You might have to push it. It's a giant ball, like a ball as big as you. Okay. So active transport always requires transporter proteins and also energy, usually in the form of ATP. Okay. So here's one example. Your book doesn't go into this much, but I'm going to go into it with you because it's important to understand different types of transport in membranes. So this is actually a transporter found mostly in eukaryotic cells, animals and plants. So it's called the sodium-potassium pump. I know most of you probably heard of that one before in introductory biology or human biology, or if you even had physiology by now. The sodium-potassium pump, or sodium-potassium ATPase, all right, it's a transporter enzyme, and it splits ATP to use the ATP energy to push things against their gradient. And so what happens is the ATP binds on the cytoplasmic side. So this is the cytoplasm, and this is the extracellular environment out here, right? So inside the cell, there it is, outside the cell. White on white is not the easiest to see. But ATP binds, and then 3 sodiums bind here. And then the ATP splits. And when ATP splits, it splits into inorganic phosphate, which you might remember is PO4 with 3 negatives, all right? And adenosine diphosphate. So it's got 2 phosphates stuck on it still. All right. So it still has some energy, but that releases a whole lot of energy. And what that energy does is it causes the protein chains to rearrange, and it pushes those 3 sodiums out. And at the same time, the extracellular surface of the protein binds to 2 potassiums. All right. Now the phosphate that was split off, it stays bound to the protein, and that phosphate bond, this still has some energy in it. Okay. And so the potassiums bind, and then the phosphate splits off, and that releases some more energy. So it's like a 2-step energy release. It releases some energy. When ATP first splits, it splits by hydrolysis. I think we talked about that before already. Water adds to it and releases that 3rd phosphate. Okay. And now we're going to release the 3rd phosphate is stuck to the protein, and we'll release it from the protein. That releases a little bit more energy. So first, we use the first batch of energy to move 3 sodiums out, and we use the second batch of energy to move 2 potassiums in. Okay, so we're moving sodium out against its gradient and potassium in against its gradient, because it's high potassium inside the cell and low sodium. Whereas outside the cell, there's high sodium outside the cell and low potassium, right? So sodium and potassium are both being moved against their gradient in a 2-step energy release from ATP, but you need ATP energy to do it, all right? And technically, if you remember your transports, this is called primary active transport, because you're directly using ATP to power your pump, all right? So it's called the sodium-potassium ATPase, or the sodium-potassium pump, and it's a counter exchanger. Another type of pump you could use is also primary active transport, is using ATP energy for a proton pump. So ATP can be split, and its energy causes the shape of a protein that moves protons from an area of low proton concentration to an area of high proton concentration. All right, and I totally can't spell for some reason, but that's okay. Okay, yeah, high. Okay, cool. So that's active transport. There's also secondary active transport, which involves basically you make an ion gradient like you are here, like put lots of sodium out, but then when the ion gradient that you've created by using ATP indirectly it releases its energy by coming back in, that can be used to push something else against this gradient. So one gradient, a proton gradient or a sodium gradient, okay, or even a potassium gradient, can be released going back down its gradient, but by something else going back down its gradient, it can pull something else against its gradient, right? Because releasing your gradient is also releasing energy, right? So two sources of energy we've seen so far: splitting of ATP and movement down a concentration gradient, especially if it's ions, because ions, not only are they moving down their concentration gradient passively, but they actually repel each other. So they aggressively move down their gradient, like, get out of the way, I'm going to go now. Ah, right. So that's your ion gradient. So ion gradients are very aggressive. And because it's charge moving, it also creates an extra level of energy. It's sort of electrical sparks. We call them electrochemical gradients. There's also group translocation. That's kind of unique to prokaryotes. All right. Yes, it is unique to prokaryotes. There, I wrote it. All right. So we have a transporter protein. You also have an energy source called phosphoenol pyruvate. Okay. So phosphoenol pyruvate is a short organic molecule, and it's got two phosphates on it. All right. And since it's got two phosphates, remember, phosphates don't like each other very much, so they repel each other, and so it's not very stable. And remember, if you're not very stable, you have a lot of energy. Okay. And so what phosphoenol pyruvate does is one of the phosphates breaks off and attaches to a sugar. Then you have a phosphate sugar. Okay. And this happens, so you take a sugar from outside of a cell and you move it through a transporter, like a carrier protein, and you put it inside the cell. And then once it's inside the cell, PEP comes along and gives off one of its phosphates. All right. And attaches it there. So now, once it's inside the cell, glucose, in this case, if it's glucose, glucose becomes glucose phosphate. And glucose phosphate is different. It's a different ligand than glucose was. And so it can't rebind the transporter and move back out. Remember, in passive transport, like we saw before, things would just diffuse back out. But because it gets trapped inside and it's got this extra negative or maybe even two negative charges stuck on it, it can't get back out. So the phosphate, you're consuming energy to add the phosphate to it, but that keeps the glucose inside, which is where you might need it. And that's okay, right? Even if you have to spend phosphate energy, because we'll see that glucose can give you a lot of phosphate energy in the form of a lot of ATP if you extract all the bond energy from your glucose and other sugar molecules. All right, so that's okay. And then I'm also going to mention, we're not going to talk about these in a lot of detail in this class, but there's also other bulk transport, especially in eukaryotes. Hold on. Yeah, so there's bulk transport in other in eukaryotes. So I'll just put this in. Oops, put the wrong pen. All right. Put this in brackets here. Okay, so this is in eukaryotes only, but there's what's called exocytosis, where the membrane actually rearranges and pushes things outside of a cell. And there's endocytosis, where there's some bulky food around. And you guys saw this in our microscopy images and videos, right? Of wrapping around something like a phagocyte does, can wrap around it and bring it into a cell. So you can bring big bulky things in, that's called cell eating or phagocytosis, or you can wrap around and bring in a lot of small things at once in bulk. Okay, that's called penocytosis. So this is cell eating, phagos is cell drinking. Penos means drinking, right? Phagos is eating, penos is drinking, and cytosis means cellular process. So cellular process of drinking, cellular process of eating. But that only happens in eukaryotes. Why only in eukaryotes? Because only eukaryotes have an extensive cytoskeleton beneath their membrane where they can rearrange these fibers and push the membrane around and cause it to engulf food or to eject waste the way that it needs to do for endocytosis and exocytosis. All right. So it's all about using ATP and also another energy source called GTP to rearrange the cytoskeleton inside of eukaryotic cells. But remember, or at least you will see in a moment, that prokaryotic cells don't have much cytoskeleton. They have a little bit, not much. just a tiny bit. All right, so your blue book question, your class journal question for this part is, give me two major differences between active transport and passive transport. Clue, how much energy? What kind of energy? What direction? Haha. All right. So you should be able to give me at least three major differences. Go for it. So try to address that in your discussion questions in online. All right. The cytoplasm. The cytoplasm is just all the fluid stuff inside there. But there's a lot of solutes that are dissolved, so it's kind of thick and viscous. So the cytoplasm, it's really, in truth, a gel. All right. So gels are, well, watery aqueous gels, like our cytoplasm and like the agar that we use for our media in the laboratory. They're hydrophilic gels, so it's lots of solutes and often fibers that form a matrix. Excuse me, and water surrounds them and forms a fluid phase around the fibers or dissolved substances that are usually a little more stationary and non-moving. Okay, but the gel of a cytoplasm, I mean, there's lots of water around the nucleoid, which is just a giant piece of DNA, giant for a prokaryotic cell, not nearly as big as the pieces in a eukaryotic cell. There's lots of ribosomes everywhere. There's lots of dissolved amino acids, and there's proteins, and there's RNA and DNA floating around, and mostly RNA. But basically, yeah, there's just a big, thick, viscous suspension in there. Okay, that's the cytoplasm. And then there's the nuclear area, which we actually call the nucleoid, which is a complex of DNA, the double helix of DNA, but it's also bound and sort of coiled tightly around nuclear or DNA binding proteins. Okay, and what charge is DNA? Sugar phosphate backbone. DNA is mainly negative, right? Because all those phosphates. Okay, so guess what charge most of the DNA binding proteins are? Guess what amino acids they have? Yeah, they have lots of positively charged ones like lysines and histidines and arginines, right? Stuff like that. So that's the nucleoid where DNA and its associated proteins are. And that is the chromosome. And in prokaryotes, their chromosome is in the cytoplasm. In eukaryotes, it's in its own membrane-bound compartment called the nuclear envelope. Okay? But yeah, in both cases, the chromosome it's called chromosome because they were first seen when staining cells, and they were little colored bodies, chromosoma, which means colored bodies, right? So the nucleoid is kind of like a nucleus because it's where the DNA is, but it doesn't actually have its own compartment. So it's like a nucleus, but not quite. But it's about, yeah, by mass, they're about half DNA and half protein. Lots of proteins to make it compact and fit in there. DNA is huge, so it has to be compacted and coiled up tightly to fit inside of a cell. All right, the ribosomes. So ribosomes are, in fact, the protein factories. How important are proteins? Crucial, right? And ribosomes they last for a little while, but not super long, so they have to be made over and over and over again. So you're always making ribosomes. And ribosomes by mass, they're about 1/2 RNA and about 1/2 DNA. But we're going to see when we start looking more at ribosome function in like 3 or 4 chapters, the RNA is actually the catalytic part. Okay, so RNA is the enzyme part, and so RNA enzymes we call ribozymes. I think I might have mentioned that before, but we'll see it again. But yeah, so ribosomes are ribozymes, right? They're RNA enzymes. So there's lots of little pieces. Like I said, by mass, they're about half RNA and what am I saying? Half RNA and half protein. Sorry about that. That's probably confusing. Half protein. Okay, so there's a small subunit, the 30S subunit. So S stands for Svedberg, this European scientist who discovered how to measure them. And what he did was he took ribosomes and he took other cell components and he put them in a centrifuge tube and he spun them around very, very, very fast. And anything really dense goes to the bottom. Less dense things are up here somewhere. Even less dense things are here, and really buoyant or non-dense things are up here. And the 30S is somewhere around here, right? Um, so S is also called the sedimentation. So does it sediment? Does it go to the bottom? Right. The 30S is somewhere there. The 50S is somewhere there when they're separated. So there's a small subunit which has it tells you how dense it is. It's not mass. Okay, this is density. I want you to understand that. So the 30S is fairly dense. The 50S is much denser, and then when you combine them into a combined ribosome, complete ribosome, they are 70S. See, it's not linear mathematical. Okay, it's density, it's not mass. And so say the 70S would be really close to the bottom, something like that. Okay, so those are sedimentation units. But you need both the large subunit and the small subunit. And what they do is they work together to bind to what are called messenger RNAs and read the genetic code that came originally from your DNA. So DNA is used to make RNA, which is the message, and the message in the RNA is read by the ribosome and by some other RNAs that we'll talk about to pump out brand new proteins, all right, based on the genetic sequence. Pretty cool, huh? Yeah, so there's a genetic code, and we're going to go through it. You guys are going to learn how to read the genetic code. Isn't that cool? All right. So the final thing I'll just go through really quickly because we're not really going to talk about these much in this class, but there are some intracellular structures that are besides ribosomes that are kind of interesting in prokaryotic cells. So in these prokaryotic cells, there's little reflective granules that give off different colors, metachromatic they're called. It's called volutin. I don't know why it has that name, but this is like phosphate salts that are stored there. So you have extra phosphate for making nucleic acids and nucleotides like ATP and GTP and TTP and CTP, right? All the triphosphates that we use to make our DNAs and our RNAs. Then you can have polysaccharide granules like starch or glycogen like granules, and that stores carbohydrate energy. And carbohydrates, all right. Carbohydrates have lots of energy. You guys know that, and you'll learn even more in the next chapter. You can also have little lipid droplets. Remember, lipids form little droplets that are excluded by water, and that's to store fat or oil energy. There's also sulfur compounds that form little granules. So for energy, those can be used for energy by certain microbes. Can use sulfur compounds as sources of electron energy. We'll see that later. Now there's carboxysomes, right? These guys are cool, and that word probably doesn't mean much to you, but they're called carboxysomes because they store an enzyme called ribulose 1,5. It's actually called bisphosphate. That's a biochemical misnomer there. Bisphosphate. Bis means there's two phosphates like diphosphate is, but it means they're on different carbons. If they're on the same carbon, it's diphosphate. If they're on different carbons, it's bisphosphate. Anyway, that's why it's adenosine diphosphate because the two phosphates are on the same carbon, right? They're linked to each other. Carboxylase. All right. And so what this does is this enzyme uses the biochemical energy from light to take carbons from the atmosphere in the form of carbon dioxide, which is completely oxidized and non-energetic carbon, and put them together to make polycarbon compounds, which have lots and lots of energy. So that's how photosynthesis works. Okay, so this is the carbon fixation enzyme. All right, the carbon fixation is the main activity that happens inside of photosynthetic cells. Carbon fixation. I don't know why my pen likes misbehaving like that. Carbon fixation. Fixation. There we go. Right, carbon fixation. All right, I'll jump down here. There's also magnetosomes. Magnetosomes have iron oxide, all right, and these help break down. I probably need a different color. Iron oxide. These are magnetosomes here, and they're drawn towards the center of the Earth. And that way, so structures like this are in a lot of marine microbes, and it tells them that the atmosphere is that way. So up that way is oxygen if they need it, and light if they need it. Okay, but what they also do is these iron oxides, they help destroy peroxides. Okay, which peroxides, here's what a peroxide looks like. Peroxide means that an oxygen is linked to an oxygen in the middle of the molecule. The problem with this is that oxygens like to fight over their electrons, and like any war, nobody wins. Okay, let me get rid of that line again. And what you get is a hydroxyl. It's called a hydroxyl radical. It's got a free radical, so it's got an unpaired electron, and you get two of them. Okay, hydroxyl radical. And free radicals, they rip electrons from other molecules, but now those other molecules have unpaired electrons because they only took an odd number of electrons, and they become free radicals. So free radicals do horrible things like rip the heads off your phosphates, off your phospholipids, decapitate your phospholipids. They don't form bilayers anymore. You lose your membranes. They damage and denature proteins by damaging the amino acid side chain, so they destroy tertiary structure, which causes denaturing, unfolding of your proteins. If you denature a protein, if you lose its structure, you lose its function. Okay, and it also causes damage. It rips the bases off of your DNA, and so your DNA sequence gets messed up, and which causes mutations. And your DNA encodes for your proteins, so your genes and your cellular activities just get really, really messed up, so they die. All right. So let me jump back real quick for a moment. I just want to talk about these for a second. This is just a little fun side story for me, because when I was in my second year of graduate school, we had to do some research and develop a research plan, just a hypothetical research plan of something that we might be interested in doing. And I was doing some research. So I was looking about membrane structure and function. I came across these things called gas vacuoles. I'm like, well, that sounds interesting. Then I found out that the gas vacuoles are in prokaryotes. They're in bacteria and they're in archaea. And what they are is they're not like a membrane vacuole like you have in a plant cell where it stores a lot of fluid. It's actually they're little pockets of protein, right? So you have these hydrophobic proteins, and they put together these hydrophobic proteins, and these hydrophobic proteins start assembling, right? And they assemble like this little cap, kind of like this, all right, like a little cone. Then they start assembling more, and they start assembling into a cylinder below the cone. And eventually, you get these hollow structures that look like this. They're hydrophobic proteins. That's why they just they assemble spontaneously. Remember that word? Just like hydrophobic phospholipids do. But air, gases can move in to this hollow space, and they do. So oxygen and CO2 and nitrogen gas from the atmosphere, they all move in, and they're filled with gas inside. So that's why they're called gas vacuoles. Okay, so the little bubbles of air. All right, and you see this? This is a cross section of all these little gas vacuoles. You see all these little bubbly things here, and you can kind of see the elongated form a little bit better down here, for example. So this is more of a sideways view. But what it does is it decreases the density of the cells. And if you have these microscopic little gas bubbles, essentially, inside the protein vacuoles in your cells, you're going to float. So if you need, especially if you need light, right? So the bacteria that I was researching actually did, they actually were did photooxidation, which means they use light energy to make ATP. They would actually move to the surface, and so they could just float to the top without having to spend much energy because they just kind of passively they're buoyant. All right, instead of having to swim up there with flagella, which requires a lot of ATP energy and a lot of proton pumping, which is also requires a lot of ATP energy. So pretty cool. So microbes, even the simplest prokaryotic cells, have some pretty fascinating molecular structures inside of them, and I thought those were pretty cool. And I passed, so I'm okay. All right, so the last main structure that we're going to talk about with the prokaryotes is a very, very important one. All right, they're called endospores. So spores are sort of thick-walled structures, dormant structures. So the cells or tissues inside are essentially asleep, dormant. Right, they're latent. Um, they're not awake, right? They're hibernating to some extent. But endospores are actually inside of the cell, and that's unique to bacteria. But endospores are also the most resistant, the most durable biological structures known. All right, truly biological structures, cellular structures. So they're resting or dormant cells. And the spore coat, which surrounds the cytoplasm and the DNA and all the proteins inside and everything, it's basically a thick cell wall, and it makes them resistant to desiccation. You guys know that word, desiccation, drying out. That's what it means. They're resistant to drying out. They're resistant to heat damage, chemicals. They're resistant to radiation, especially ultraviolet radiation. They're pretty resistant to it. But one of the interesting components, and it's not even, I don't think it's even the major component. The main component is actually peptidoglycan. It's just like a really thick peptidoglycan layer. But it also has this interesting ionic polymer inside of it. It's called dipicolinic acid. So here's dipicolinic acid. See, it's got a carboxylic acid with a negative charge. Carboxylic acid with a negative charge. And the middle of it is it's a conjugated double bond ring. I might have mentioned those to you guys before, but which means it's very stable. But it's got nitrogen here, so it's partially, partially negative actually. Is that right? Yeah, partially negative right there in the middle. All right, but it's got the negative charges here. But so it has these stable ring structures, and then a negative charge at each end. Stable ring in the middle, and a negative charge at each end. So we have carboxy group, and they get chained together by calcium, because calcium has two positive charges, so it can balance. So it basically forms an ionic polymer, kind of like this. Boop, boop, negative, negative, and then we got calcium with two positives, right? So like this, negative, 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 and then in the middle, you throw in a calcium with its two positive charges. All right. So the calcium makes a little cationic bridge between these anionic organic structures. So so it's a very unique substance, and we don't really know, I think we really know structurally exactly how it works. But I'm guessing that these ring structures, usually when you have these stable aromatic ring structures, which means they have those conjugated double bonds that are shared around a very stable ring, they're good at absorbing energy, because those types of structures are found in pigments like chlorophyll and carotene, things like that. Um, and so it's probably good at absorbing ultraviolet energy or something like that. So it might help protect the interior of the spore, because this is part of the spore coat, which is just a super dense cell wall. So the two main species that we'll talk about in this class that have it, there's Clostridium. We'll talk about some Clostridium species later on, and Bacillus, which we've already talked about and even used a little bit in laboratory before. So they make endospores. So all known endospore formers are in fact gram positive. Okay, keep that in mind. All right, so the word sporulation just means endospore formation. So if it's sporulating, it's making endospores. They kind of make endospores just randomly, but if you stress them out, they make more endospores. Does that make sense? Because they're afraid the cell might die, so they need a new cell that's extra protected, like a little escape pod. So let's see how that happens. Yeah, and so they form a spore, which is a little cell inside of a big cell coat, and then when they germinate, the cell inside digests the spore coat and starts to grow out and divide more actively, right, and so on and so forth. That's vegetative state, which means actively growing and metabolizing. And so that's called germination, just like when a seed breaks out of its seed coat, right? Except a seed has a whole embryo inside. Endospores just have one little prokaryotic cell inside. All right, so here we are again. So we have our cell, and it goes through replication of its DNA. So it has two copies of its chromosome, but instead of dividing right down the middle like bacterial cells usually do, it doesn't do that. It divides asymmetrically. All right, so not down the middle. Asymmetric division. Um, and then not only does the cell wall kind of pinch off in the middle, but the plasma membrane pinches off in the middle. All right. And so what you end up with is you had a mother cell, and now you have two daughter cells, 1, 2, where the first daughter cell is very small, and the second daughter cell is sort of your normal vegetative cell. But daughter number 2 basically becomes the home. This is the home girl or the home daughter of cell number one. So cell number one, the daughter cell's second daughter cell's membrane pinches off around it, and then that space between the two membranes fills with this brown peptidoglycan. Okay, then around the peptidoglycan, there are spore coat proteins as well as dipycalinic acid. Dipycalinic acid, these are the AN proteins, form the spore coat around the outside here. All right, this kind of thick spore coat. Now the daughter cell, the big sister, she does fine until she dies or she gets damaged. Then if she dies, then this little sister is dormant, asleep, and it will escape like an escape pod, right, from a from a spaceship. It's like an escape pod, and this can sit there and it can sit on dry soil or concrete for years. It can be frozen in the Antarctic. It can even be fossilized um, in deep into the Earth's crust. Scientists have found deep thousands of thousands of years, thousands of layers and years deep into glaciers have found frozen endospores. Deep into the Earth's crust, millions of years deep, they have found um, endosporulating bacteria. They brought them out, they put them in a rich broth medium, and they germinated. They survived hundreds of thousands to even millions of years. Whoa. All right. So here's the endospore. So here's the spore coat around it. All right. And the spore coat there is not just empty, but but here's the endospore. Okay. Um, so pretty cool, huh? But yeah, these are the most durable. Like it's really, really hard to kill them. All right, and in fact, the sterilization methods that we use in the laboratory, they're designed to kill endospores. Because if you can kill endospores, you can kill any kind of cell. But these are the most durable cells on the planet. So Bacillus anthracis, all Bacillus species make endospores. They're all gram-positive, and Bacillus anthracis, what does that cause? Anthrax. Who discovered this bacterium as the cause of anthrax? Robert Koch, all right? Remember him? Koch's postulates. So make sure you review those too. Yeah, so these are just, yeah, endospores are cool. Kind of scary, but cool.